Hello, this is Venkatesh Balan, a faculty in College of Technology. We are focusing on producing biofuels and biochemicals using microalgae. Okay, so why microalgae? Because we are trying to use algae to fix the carbon dioxide from the atmosphere. You heard about greenhouse gases. We are trying to fix those carbon dioxide using this algae, and we can produce, using those algae, we can produce fuel molecules like biodiesel and other kind of molecules, chemical molecules from this algae. So what we do is, typically these algae are found in the wild. So we try to culture them on the plates first. They grow them, we can see the streaks on the plates. And then once you grow them, we just pick up a small colony from this plate, and eventually we try to inoculate the media. So basically, like what we need food, the algae also need food. We just try to give them enough food and nutrients, and they just fix the carbon dioxide and then try to grow them. So here is another example where we try to produce algae by growing them in this kind of tubes. We have two different algae here, spirulina and chlorella. So what we do is we try to add the inoculum with the algae inside and just start bubbling CO2. And we need enough sunlight and CO2. This can keep growing and it can fix the carbon dioxide and build them as a body mass. We then harvest the algae, we process them to make fuel molecules in the lab. So using enzymes and other kind of environmental friendly technologies. So here I'm going to show you a demonstration about how to take microalgae and then process them into biochemicals or sugars first, and then eventually we do fermentation to produce biochemicals. The first step we have to do is we have to take the algae, and we have taken already this algae in a dry form. We weigh them in a bottle, and then eventually once you weigh the right amount of algae in the bottle, we open this cap. We test them in small scale, like 20 ml scale. So we add 20 ml of water into this, and once we finish adding the water, we use industrial enzymes. These are all commercial enzymes produced by using fungus in the industry. So what we do is we just take those enzymes, put them in a smaller tubes like this, a pint of tubes. We just take a smaller amount, maybe microliters, using this pipetter. So we take a smaller amount, and then we just load those smaller amount into this algae solution. And once you finish adding them, we just try to close them up and then incubate this just for 2 hours. And this is good enough to hydrolyze all the algal cell wall into sugars. And same like what you are producing when you are taking starch, and when you are eating those starch, our system has enzymes to digest the starch into sugars. The same way we are doing this in the laboratory to produce sugar molecules. And that is a major focus of this lab, to convert this high concentration of sugars from renewable resources like algae, because algae is fixing carbon dioxide and then converting them into sugar molecules. Eventually we can take this, and after we finish the hydrolysis, we centrifuge and then take them and then do a fermentation using microorganisms which can convert into chemicals and fuel molecules. I'm going to show you how we grow the algae in a growth chamber. So this is a growth chamber which has a temperature control and also with all light. We are simulating a natural environment. So we try to culture the algae and produce them in the flask and keep them in the light. So sometimes we shake, sometimes we don't shake. And this algae, we put all the nutrients in the flask and just needs the light and just keep consuming the carbon dioxide. We also externally supply CO2 into this chamber, and then it can continue to grow the algae.